So our first talk is from Jacques Dutrieu, who'll talk about modified interferon alpha subtypes and chemokine networks in the thymus. Jacques.
Thank you. Good afternoon. I will present you the work I've done in Rémi Chenier's lab at the Institut Cochin in Paris, and which was focused on the impact of HIV infection on thymopoiesis. HIV infection is characterized in humans by an affected thymopoiesis due to a diminished thymocyte proliferation. Several indications point to the fact that interferon alpha response could be at the origin of this phenomenon. In both human and macaques, plasma interferon, there is a peak of plasmatic interferon alpha around day 10 post-infection, just before the peak of viral load in humans and macaques. In our study, 24 Chinese rhesus macaques were infected using the SIV MAC 251 strain. Blood was sampled every 2 or 3 days, and we monitor the thymic function, the T-cell subset dynamic within the blood, and we also measure the plasmatic interferon alpha concentration. In parallel, necropsies were done on healthy macaques and in acute infected macaques at day 3, 7, 10, and 14. The thymus was sampled, and interferon alpha and chemokine mRNA were quantified. As you can see on this slide, by day 10 post-infection, there is a decrease in intrathymic proliferation, and this diminished intrathymic proliferation correlates with the amplitude of the interferon alpha plasmatic peak. As you may know, interferon alpha is a diverse family, which is composed of different subtypes. For example, in humans, there are 13 subtypes, and in our model, there are also 13 subtypes. This diversity is conserved during evolutions in mammals. We assess the interferon alpha subtypes production within the thymus in uninfected macaques and in infected animals. Each X represents one interferon alpha subtypes, and each color an animal. As you can see, in uninfected animals, there are very low number of interferon alpha subtypes which are produced. And it's heterogeneous between the different animals. During acute SIV infection, there is a diversification of interferon alpha response within the thymus, and also an homogenization between the animals. Thus, acute SIV infection is characterized within the thymus with a diversified thymic interferon alpha expression. To see if these interferon alpha subtypes could have an impact on the thymocytes differentiation, we've done an in vitro differentiation of simian thymocytes test in presence or in absence of the 12 interferon alpha subtypes. After 14 days of culture of the sorting immature thymocytes, we assess the frequency of mature cells and also their historic of proliferation using a CFSE assay. As you can see, without interferon alpha in the medium, thymocytes extensively proliferate by day 5 post-culture. We can class the interferon alpha subtypes into 3 families. First, the interferon alpha 1, 6, and 14 has a very light effect on thymocyte proliferation. Interferon alpha 5, 8, 9, and 13 has a moderate effect on thymocyte proliferation in vitro, whereas interferon alpha 2, 3, 4, 7, 12, and 15 had a very big impact on thymocyte proliferation in vitro. Thus, interferon alpha subtype differentially inhibit thymocyte proliferation, and I didn't show you because of lack of time, without affecting their differentiation capability. Furthermore, in vivo produced interferon alpha demonstrate a major antiproliferative effect. As you may know, thymopoiesis is driven by chemokine within the thymus. In mice, it is known that CCL25 drives a cortical migration of thymocytes after they enter in the thymus. CXCL12 is responsible for their retention within the subcapsular region, where they extensively proliferate after the beta selection. And finally, CCL19 is known in mice to be one of the most chemokine which is implied in T-cell egress. To know if these chemokines were expressed within the same compartment in the thymus, we assessed in situ hybridization, and as you can see on the right panel, CCL25 appears here in blue, is only expressed in the cortex as in mice. We quantify the mRNA expression of this chemokine within the thymus after infection, and you can see that there is a significant increase of the transcription of CCL25 by day 3 post-infection. You can see also that CCL19 is, as in mice, expressed only in the medulla of the thymus. It is also increased by day 3 post-infection. CXCL12 is mostly expressed in the subcapsular region, as in mice. And the transcription of this chemokine is decreased by day 3 post-infection within the thymus. I showed you that during acute SIV infection, there is a diversified interferon alpha response, and this interferon alpha can inhibit thymocyte proliferation. Furthermore, there is a very big modified thymic chemokine network by day 3 post-infection. All these results point to an accelerated thymopoiesis, which follow the model I will propose you. First, the increased transcription of CCL25 will allow a faster migration within the cortex of the thymocytes. Decrease of CXCL12 and of thymocyte proliferation due to interferon alpha will allow less retention within the subcapsular region. And finally, the increased CCL19 transcription will allow a faster thymic T-cell egress from the thymus. Acute SIV infection results, though, in an accelerated thymopoiesis, which will transiently increase the thymic function. These two phenomena, will imply an increased naive T-cell diversity just after the virus entry within the organism. This phenomenon could eventually lead to the characteristic HIV-associated thymic exhaustion, which is observed in chronically infected patients. I would like to thank Rémi Chénier and all the members of his lab, and also our collaborators and funders. I would like also to acknowledge the organizers for giving me the opportunity to present my PhD work here, and also the International AIDS Society for providing me a scholarship to come behind you today.
Time for a couple of quick questions. Martin?
Martin French, University of Western Australia. Have you examined the role of interleukin-7 in your system here?
Well, in the in vitro system, you mean?
Yeah.
Yeah. We all the in vitro system was done with 5 micrograms of IL-7 and FLT3 ligand. But we haven't changed the concentration of IL-7.
The reason I ask is because we've got a paper in press at the moment in the Journal of Immunology where we've looked at peripheral blood T cells and shown that interferon alpha has quite a profound effect on inhibiting the proliferative effect of interleukin-7.
Yeah.
And I think it would be interesting for you to do that in your system.
Of course, we have looked at the level of interleukin-7 in our macaques too, and there is no change within the time post-infection in the thymus, but in the blood there is a slight increase in the quantity of IL-7. Of course, it could be very interesting to look at that because of the two pathways which lead to P27, KEAP1. Yeah, of course.
Have you or others examined thymopoiesis in other acute infections? Is this characteristic of HIV? To me, this could just be what happens.
Of course. It is known that in measles acute infection in children or in macaques, there is this kind of phenomenon like an enhanced thymic function. We also saw it, but it's very transitory in patients which were vaccinated with yellow fever.
Hi, nice work, Jan van Winsen from Hamburg. Actually, do you had the chance to look in the peripheral blood of these animals and the presence of recent thymic emigrants and how that correlates?
Yeah, we looked at recent thymic emigrants, of course, which were CD31 high. There is a decrease in CD31 plus cells within the blood, but a rebound after day 10 post-infection, which is very slight. And we think that this rebound could be also the, could be, oh sorry, my English is horrible.
You can say it in French.
No, this could be the thymic, the T cell egress from the thymus, but also the comeback from the homing in the organs.
Okay, and did that correlate with viral replication or viremia?
No.
Thank you.
So, if there's no more questions, thank you for a very nice presentation.